this is a B6 triple science booster pack. So this video works well after watching the B6 in a nutshell, which is all the double content, and then you can use this to boost up for the triple content. First thing we're going to look at, we talked about asexual and sexual reproduction. Now we're looking at the advantages of each. So sexual reproduction, this is the one where there is fusion of gametes. So it produces variety in the offspring because the genes are coming from both of the parents. If the environment changes and we get some mutations and we get some features that are better, then natural selection can occur. And selective breeding using sexual reproduction can increase food production. Advantages of asexual reproduction is that because only one parent is needed, it's more time and energy efficient. It's so therefore faster, and you can get many identical offspring. So if you've got great features of an offspring, then you can get lots of them that all have those great features. There are some individuals that can use asexual or sexual reproduction depending on what the situation is like. So malarial parasites, okay, these were our protists in B3, and they can use asexual reproduction when they're in the human host. If you remember, they go off to the liver cells and they reproduce in those. When they're on the mosquito, they use sexual reproduction. Fungi, fungal spores, they can use asexual reproduction using those fungal spores, and sexual reproduction gives variation. Plants, we've got the strawberry plant, which doesn't produce any flowers. It's got runners, so uses asexual reproduction, again like the spider plant, and yet you can also use sexual reproduction using seeds. That's gives us our different examples. Now let's have a look at the structure of DNA in a bit more detail. So we know that it's a polymer. We know that it's these, it's got like this backbone and it's rungs of a ladder. Okay, so we've got this backbone which doesn't contain any DNA, any genetic information, of phosphate and sugar alternating. Okay, and then we've got it going this way. And if you notice, you know, just for being exact, we can see that this one's actually upside down. Okay, so we've got phosphate, sugar. Phosphate is always the circle, sugar is always the pentagon. Okay, and attached to the sugar we have a base, and we know that there are 4 different bases. Now when we take one phosphate, one sugar and one base, we call that a nucleotide. Okay, and if we take one nucleotide, 2, 3, 4, 5, 6, that is 1 amino acid. Lots of amino acids together make a protein. So let's have a look at that in a bit more detail. So we've got our nucleotide, like I said, phosphate, sugar and a base, and those bases have corresponding bases which they match up to, and they're held together with hydrogen bonds. Okay, so A always matches with T, C always matches with G. I tend to remember the C and G because they both have that curved shape. Okay, the curve of the C, the curve of the G. Okay, you do not need to memorize these names here, so just A goes with T and C goes with G, held together with those hydrogen bonds. Here's some examples then. So when we get different 3 lots of base pairs together, we get an amino acid. We've got a different combination. Sometimes they're the other way round. All of these different combinations give a different amino acid. Okay, ensure a different amino acid is made. Put lots of amino acids together and we've got our protein. Okay, so all those different combinations can make lots and lots of different amino acids. Moving on to protein synthesis. Now, we learned way back, ages and ages ago, we learned that protein synthesis occurs at the ribosomes. We also know that we eat our protein, we digest it into amino acids, and then we know that somehow those amino acids make proteins again. Now, we're over here in the nucleus. In the nucleus, temporarily, the DNA unzips, and we can see it's unzipped here and here. Some of these bits of amino acids that have been drifting around have diffused into there, and they have created, by matching up using those hydrogen bonds, they have created a template. That template then removes itself from the DNA. The DNA apologies. The DNA then zips back up on itself. The template leaves the nucleus and it drifts its way over to the ribosomes. Here at the ribosomes, we have these molecules called carrier molecules. Here they are, carrier molecules, and they bring the corresponding bases over to that template to build up that protein chain. Let's have a look at that written out. So DNA in the nucleus unzips. Amino acids match up to those bases on the DNA, making a template. The template drifts over to the ribosomes. In the meantime, that DNA zips back up again. Over at the ribosomes, carrier molecules bring those other parts of the amino acid to join up to build that long protein chain. When the protein chain is complete, it folds up to form a unique shape. The unique shape enables the proteins to do their jobs, and some examples of those proteins are enzymes, hormones, and forming structures in the body, such as collagen. Okay. Let's have a look at mutations that can occur. Now, we know that mutations occur all the time. Sometimes, just like a spelling mistake, we can still figure out what the word is. They make no difference at all. So most don't alter the actual protein and how it functions. Sometimes those mutations will be significant enough that there may be a change. It might be that the enzyme no longer fits the substrate into it, the active site maybe changes a bit, or it might be that genes switch on and off. So basically, if we have a look at this picture at the bottom, we have a section of DNA, we've got a gene there. We've got a non-coding part of DNA and we have a coding part of the DNA. The coding part of the DNA dictates what the protein is going to look like. If there is a mutation in the coding part, then it could be that that enzyme no longer fits into the active site, is no longer appropriate for the substrate to fit. Okay, so the lock and key theory is not able to work. It could be that there's a mutation in the non-coding part. And this is where, when we watched Animal Farm, we saw that fish that should have had their switch of growth turned off when they get into particularly cold water, and the switch was broken because there was a mutation in the non-coding part of the DNA, so they kept on growing and growing. Okay, so mutations in different parts of the gene will affect how that responds to that situation. Okay, let's go to cloning. So cloning is really simple. We can do a simple cutting from a plant and we can put it into some of that rooting powder, the auxin, we talked about that at the end of B5, and then we can put that into a pot, allow it to grow, and we have a very cheap clone of the original. Embryo transplantation. Okay, this one is one that people tend to forget, not because it's difficult, I just think there's not many questions on it, so people don't keep it in their mind. This starts off with selective breeding. So we take the two individuals that we think are going to produce a really good offspring. Now, selective breeding, we would just put them together, allow them to mate, and then they would have the offspring, hopefully with the desired characteristics, and that would continue over generations. That's what we said in selective breeding. In embryo transplantation, we take those two individuals that we think are going to have the good features, and we do IVF. So we take the egg, we take the sperm, we fertilise them together in a petri dish. Now, if that was going to be identical twins, when it turns into an embryo and it starts going through mitosis, if that embryo splits, we get identical twins. Now, what they actually do is they force that to split up to 4 times. So you can see that we've got our 4 individuals at the bottom. Each of those embryos is implanted into a surrogate, into the uterus, and then when the cows give birth, they are all clones of each other. That's embryo transplantation. Think of it as embryos because it's choosing the individuals and they haven't yet existed. Then we go on to adult cloning. This is where the individual already exists. We can see it's got great features and we want another one just like it with exactly the same DNA. So the DNA of this and the DNA of this are exactly the same. What we've done is we've taken from this sheep here, we've taken some donor cells, probably body cells because they're really simple. They haven't differentiated very much. And here we can see we've taken them. From another sheep, we have taken an egg. Why? Because we need that egg cell. It's going to turn into an embryo. And embryos allow us to get all of the different cells that we need. But we don't want the genetic information in this egg cell. So we remove this nucleus. We fuse them together using an electric shock. That starts mitosis and the embryo forms. When they've looked at that embryo and they're happy that it's developing in the way you'd expect, that is then put back into the surrogate's uterus. Really important that you say where it goes into the uterus. And then eventually we get the cloned lamb. It's genetically identical to the original sheep that we took the body cell from. We talked about Darwin already. We talked about Darwin having this natural selection. He called it survival of the fittest. And this was related to mutations. So he said that individuals produce genetically diverse offspring. The offspring compete for food, water, mates, and have to avoid predators. Now, there are differences because it's sexual reproduction and because of mutation. So the offspring with the fewest competitive traits, the poor traits, they die off. The ones that are really competitive, survival of the fittest, so they're the fittest, they've got the best traits, will live longer, reproduce and pass on those features to their offspring. Now we also looked at Lamarck. Lamarck was the guy who said, whatever changes happen to you in your life, you will pass on to your offspring. This is like when he said, this is like him saying, if you get a tattoo, then your child will be born with a tattoo. Obviously that's not the case. So Darwin's idea was about genetics, Lamarck's idea was about the environmental changes that occur to you. So Lamarck's theory says that changes that occur in an organism can be inherited. So whatever happened to you in life will affect your offspring. Evolution occurs by inheritance of acquired characteristics. His theory is unlikely because acquired characteristics do not affect our genes. Whereas Darwin's theory said evolution occurs by natural selection. Genes which give an organism a competitive advantage will be selected because the other ones will die out. And acquired characteristics cannot be inherited. Now, Darwin's theory of evolution took a long time to be accepted. This was because the theory that he gave challenged the idea that God made all animals and plants that live on Earth. There was also insufficient evidence at the time, his theory was published, so he didn't have that kind of backup of genetics. He just had observations where he did all of his work with the finches, and we didn't understand the mechanism of inheritance and variation until 50 years after his book was published. In 1859 he published his book Origin of the Species, and this picture up here was a picture that was in the paper that was kind of poking fun at Darwin, saying how can he believe that we evolved from primates. Okay, going on to Wallace. Now, Wallace worked with Darwin and he did lots and lots of different things. So he actually did some work on natural selection with Darwin. He published, anyway, he helped Darwin publish his book, The Origin of the Species, which Darwin was famous for. He also worked on evolutionary theory and warning coloration in animals. So this was where it's, animals may display certain colours to ward off predators or to encourage mates. And then he did stuff on speciation as well. Okay, and so he was kind of more known for the speciation stuff than anything, but he did work with Darwin. Now let's have a look at what speciation is. So this was where we had our single ancestral interbreeding population. We have, and you can see this works as a 6 marker. We've got the geographical isolation splits the population. Maybe they've gone to different islands in the Galapagos, for example. The conditions in those different geographical areas are different. So then we get the natural selection occurring. So we get the mutations or the variation, and the beneficial ones live longer, reproduce and pass it on to their offspring. Then, as that over hundreds of years, we see those populations evolving differently. Eventually, if we bring those separate populations back together and they interbreed, and that is unsuccessful, meaning that the offspring that are produced are infertile, can't have offspring of their own, then we can categorically say they have become separate species. And we call that whole process speciation. Final thing to talk about, last slide. This was talking about a Mendel. Now, Mendel was doing work at a similar time to Darwin. He was a monk. He was just interested, walking around his garden and seeing that some pea plants produce smooth peas and some produce wrinkly peas. And what was really good about the experiments he did is he got the results very quickly because he was working with peas, so there was a quick reproduction, and he was able to do this on a large scale, so he got more reliable results. What he did is he pollinated the pea plants using a paintbrush. So he was able to say, okay, I've got a smooth pea plant, I'm going to pollinate it with a wrinkly pea plant, and I'm going to see what offspring are produced. From this, he understood about dominant traits, recessive traits. He understood how to make Punnett squares. He was called the father of genetics because he provided the evidence that Darwin lacked, that would have supported Darwin's findings from his observations. Now, because he was a monk, he didn't really know where to publish his findings, so he published them in quite an obscure journal, and it took a long time after he died before people discovered the work he'd found. But he was able to really pioneer the work of understanding genetics and understanding the passing on of genetics to offspring. Hope you found that useful. Thank you.